Hello, class. This is cell respiration, the middle part of this huge chapter 4. And I plan on doing this in person, but I'm feeling sick. And to be cautious, next week I'll send an email, but we won't have class in person. So I'll record this lecture, the last lecture, and we'll do the practical online too. That's what we're doing. And let's do this one is going to be. We'll look at the chapters on cell metabolism. Now we're going to look at respiration, and we'll begin looking at DNA and what genes are doing too. All right, could have shaved for you, I know, but that's okay, as long as you can hear me. So we have good stuff here, and a lot of you have had this in high school or in biology in college, so we'll review it, and we're not going to get into excruciating detail, but you got to know the basics of how we survive, how we burn sugar to get ATP, which really is what powers everything that we do. So cell respiration, 3 parts to this whole process. Glycolysis is the first one. It's going to split sugar in half. That's what it means, glycolysis of sugar. So it's going to break it in half. It'll give us a little bit of ATP, a little bit of energy. And you could just do that, actually, and get a little bit of energy. But if you go on, if you have oxygen eventually, you can do this citric acid cycle and then the electron transport chain where we really can harvest that energy. And if you just do glycolysis, you can get a couple ATP for a sugar. But you get all the way through it, you get like 30. All right, so you can see the difference, how it'd be kind of wasteful to just do glycolysis. But if you don't have oxygen, you can't go all the way to the end. All right, and so some organisms are anaerobic, right? Some bacteria, the ones that cause swamp gas, that live in low oxygen conditions, are completely anaerobic. And so they do glycolysis, they get some energy, but us animals, we need oxygen. We've got to be able to do aerobic respiration in order to keep up with how expensive we are, how much energy we need. But indeed, you guys that do sprints, that do anaerobic activity, where you're such bursts of activity that you don't have enough time to catch up with your breathing, you know you can do it, but you're going to get cramps, you're going to feel pain, and so if you're going to do any kind of aerobic activity, you need to get enough oxygen to those cells. So first of all, where this takes place. Glycolysis takes place in the cytoplasm. And indeed, remember, bacteria don't even have mitochondria, right? So that wouldn't happen in there anyway. So glycolysis is going to split the glucose in half. And you get a little burst of energy. And then those 2 halves, called pyruvate, that goes into the mitochondria. And then you do the rest of it. It happens in the mitochondria. Very nice. You can see Krebs cycle or citric acid cycle. The book is using citric acid cycle, but I learned it this way. Either one's fine. All right, so that's what those mitochondria do. Remember, they're symbionts that are living inside our cells. We give them sugar, they give us ATP, everyone's happy, right? And what's amazing is that this glycolysis, you can see it has 10 distinct steps with 10 enzymes in these 10 steps. It's the same freaking steps in enzymes in all of life, from bacteria to mold to trees to puppies to you. So just so obvious how we came from a common ancestor. We inherited this, just these real basic things to life, like how you make energy. This glycolysis is just think of it as being very primitive, very necessary. So with all the changes between us and a tree, we've inherited this important step. All right, so here's some chemistry for you chemistry lovers out there. And pretty much, I'm going to break it down to really the beginning and the end and the energy that comes out of it. So you start out with glucose. You can see that has 6 sugars on it. 6 carbons, I'm sorry, it's a sugar. It has 6 carbons on it, usually in a ring like that. And what we're going to do is we're going to put some phosphates on it. We're going to phosphorylate it. So we actually use up 2 ATPs. We use energy to energize that glucose molecule. And from there, you can see it's going to split in half, right in half like that, into these two, what will eventually be these pyruvates over here. And then look at all these steps. And what's going to come out of it over here, if you see, there's 4 ATP. So you need to know the net profit off of this is 2 ATP. But you actually invest 2 early on, and you yield 4. So yeah, the net is 2. So you get a little bit of energy, but you had to spend money to make money, right? You had to put it in the beginning, and you end up with these 2 pyruvates, which are 3 carbon molecules. You start out with the 6 glucose, you end up with 2 3 carbon pyruvates. And then those will move into the mitochondria, they'll be pumped into it, and then that will be the next steps. Yeah, and so you saw that the ATP, and then you're also going to see these NADHs. You actually take NAD, and then you'll charge it up with the hydrogen. You'll put that on there to make NADH. So NADH is another electron carrier. With that H on it, it has a little more energy. It's going to carry it to the last step. Yep, and the ATP, that's your cash money. That's what you're going to use for muscles and reactions, right? The NADH is just temporarily storing energy to the last step, that electron transport chain. All right, well, without oxygen, we call it anaerobic. And you'll hear fermentation, right? Remember I made wine in the storage closets. I had grape juice, I think, and I put some yeast in it. And I just remember I had a cork. I just remember that it blew up, and there was just like this red wine along this white stuff. I didn't drink until college, really, but I guess I was a scientist early on. But anyway, maybe you guys have done similar things. That's embarrassing. So now, yeah. So we need oxygen at the end of this. That's how I want to let you know. And there are some organisms that don't. They use something different, but we need oxygen absolutely. And so without it, we do glycolysis, and we get a little bit of energy. But I'll show you in the next slide. The issue is this. If you just get energy by glycolysis, you keep making NADH. You keep making it, but it doesn't get used because the NADH you use at the last step with oxygen. Without it, as NADH builds up. And it's high energy, like you think, oh, that's good. The problem is you need the NAD, you need it to be used up to come back to get recharged in order to keep the reaction going. But I'll just tell you this, is that you can kind of get rid of it if you, we make lactic acid or lactate, and yeast make ethanol, which is nice. That's where we get. All societies have figured out how to ferment sugars to make alcohol. Mexico used agave, potatoes to make vodka, wheats to make whiskey, grapes to make wine. So any of these basic sugars, you can put yeast in there, and they produce this ethanol. Yeah. So if you use, again, you use glycolysis, you get 2. 36, that's wishful thinking, 32, 34. So just so you get the point, let's see if you guys get it here, is that you can make glucose into the pyruvates through glycolysis, and you get some energy. So you guys can run that sprint because you're getting a little bit of ATP, but you keep making NADH too, which is cool. You use that at the end to get more energy. But if nothing's going on at the end, it builds up and you use up all the NADs. And damn it, you need that to keep it going. And so what you do is you use up some of the NADH and us to make lactate. And so you get that cramping, and then once you're done with the sprints, you can breathe, and your body will convert that lactic acid back to pyruvates and use it as energy, but it takes time. You build up kind of a debt like that. And then we've learned, of course, in brewing that we can harness this ability to make ethanol. All right, produces 2 molecules of pyruvic acid. 1 6-carbon glucose to 2 3-carbon pyruvates. And what do we get in glycolysis? How much ATP net? We also get 2 NADH. All right, and so we're going to move here. We'll go through this cycle, the citric acid cycle. Realize that 2 pyruvates will come in for every glucose You broke, right? Because there's 2. Oh my God, this is the reaction. You guys, did you have a teacher that taught you this that was really a biochemist that wanted you to memorize each of these and each enzyme? Well, I'm not that teacher. All right, how's that? But what I want you to know is what comes in, what comes out, and this is a cycle. And a cycle means that the original, this oxaloacetate, this original molecule that you have, you're going to, when you burn that, when you bring in the pyruvates, some of that carbon is going to attach to this to make citric acid, and that's going to go around, and eventually it's going to make oxaloacetate again. So that's what a cycle means. When you make the same product again, and then it goes around again and again. And so when you break one glucose, this is going to go around twice because it's going to run that pyruvate, the 2 pyruvates that you made. So what do you need to know? All right, so the pyruvate is going to have how many carbons? 3. Remember, there's glucose had 6, so it's going to have 3. As soon as the pyruvate comes in, well, it's an enzyme, but you're going to get rid of a carbon dioxide. Yep, you're going to get rid of a carbon. And so you're going to end up with a 2-carbon, so acetic acid, acetate, and then coenzyme A is going to attach to that 2-carbon, and that coenzyme kind of carries it to the oxaloacetate and attaches it. All right, so maybe I lose some of you. You end up with a 3-carbon pyruvate that comes in. You lose a carbon, and it turns into carbon dioxide, which bubbles away. You get a little bit of an energy from that. And then that 2-carbon sucker, a coenzyme helps shepherd it, and it's going to be reused, this coenzyme. It's going to bring those 2-carbon to attach onto this 4-carbon oxaloacetate, and it's going to make citric acid, which is citrate right here. And from there, each step has an enzyme. Each step is going to change it, change it, change it, change it. And as you go around this whole thing, you're going to make a little bit of ATP and like 8 NADH, something called FADH2. They're both going to carry electrons for you. Yeah, and what comes out of it? All those carbons that came in, carbon dioxide. So that 6-carbon sugar, every one of those carbons bubbles away as carbon dioxide, and you breathe it out. Yep. Yep. So it comes in as sugar, and oxygen comes in. What's going to go out is energy, and then water and carbon dioxide. All right. So every carbon is broken. And this is like, you can burn wood in the fireplace, but this is just, that's kind of explosive, but this is just slowly taking the energy from these bonds and harnessing it. Either an ATP, which you use immediately, that's good stuff, or these NADH or FADH, which carries the energy to that last step where you make ATP. Yep. And so this sucker gives you 2 ATP and 8 of these electron carriers. And it's going to carry these electron, even the first step. Remember we made the NADH? Here we're making NADH and FADH. It's all going to be carried down to that last step, ETC's Electron Transport Chain. So you'll hear, and I sound like I'm kind of wishy-washy, but I mean, about 30, 32, 34, I've heard 36 sometimes, ATPs are theoretically possible from this reaction. But even like pumping in the pyruvate, everything takes a little bit of energy, so your profit is always like skimmed off. So this would be, you could get every bit of energy out. And so you need those first 2 steps in order to make those NADHs and FADHs, which are carrying energy, to that last step. And that gives you the big payday. So I love this. This is beautiful here. I mean, here metaphorically, you can think about, we don't use sugar directly to like run our muscles. Just like you go in an arcade, Orchard Beach or something, you don't use cash to run the machines. You put the cash in a machine that gives you tokens. So think about that. We transfer ATP as the token. That's what you actually use in the machine. And so beautiful. Glycolysis gives you 2 tokens. Ching, ching. And then you go into the Krebs cycle. Do the ching for you, 2 more. And then also NADH, NADH, 8 NADH, FADHs, and all of these that you've been saving up, they go through this kind of a stepwise thing where the energy is taken because it's used to run a pump that's going to give you the big payday. Yep. And if you guys hold your breath, you can do glycolysis. You can get 2 energy tokens. And over here, you guys can study this on your own, but this is more realistic instead of, we're not talking metaphorically now. The ATP, you can see, so you do need to know, glycolysis is going to give you 2 ATP and 2 NADHs. ATP is used immediately for whatever you want to use it for. Then when it goes in, that 3-carbon pyruvate loses a carbon, and it gives you a couple NADHs. And then that 2-carbon, it's now acetate, is going to bind with a coenzyme, which is going to bring it in, attach it to oxaloacetate, which is going to turn into citric acid, and it's going to go around, and it's going to give you 2 ATP. It's kind of modest, but it's going to give you 8 of these electron carriers. And all the electron carriers are going to that last step, the electron transport chain. And as long as there's oxygen at the end, it's going to run. As soon as, like, unplugging a plug, if you take away the oxygen, this all backs up, and you can only do glycolysis. Yeah. All right. And again, location's important. Glycolysis is in the blue. It's out on the cytoplasm. The rest is in the mitochondria. All right, now we're to the last step. And the cycle, it happens in the mitochondria. So all the enzymes are there, right? And it goes through all these intermediate chemicals, and carbon dioxide's bubbling up, and electron carriers are diffusing down. So it's amazing to think about. It's all happening, you know, simultaneously. But the electron transport chain, it's a bunch of important enzymes that are studying that inner membrane of the mitochondria. Remember I talked about an outer garbage sac, and then, not a garbage sac, I talked about a grocery plastic bag. Imagine crumpling up a Hannaford bag and putting it in another bag. And that surface area in there is tremendous. It's bigger than the surface area of the cell. It's just really convoluted in there. And all along there, you've got all these enzymes. And those electron carriers come in, and it's like electricity. It goes through these, and it's going to cause a pump. It's going to pump hydrogens, and that's going to make your ATP. And that's what it's doing. It's pumping hydrogen ions, which is a proton. So some here called a proton pump. It's a hydrogen ion, right? Because there's no electron, H+. And you need oxygen. Oxygen does not give you any energy, but you need it in order to suck out the energy from that sugar. You need the last step, because oxygen is going to combine with that last electron. It's going to take it. It makes water. So water is the product, but you need that oxygen. I love this animation, and this molecule is called ATP synthase, so it makes ATP, and it actually rotates. And there's cool research how they found this out. They put kind of glow in the dark molecule on top of it, and then they put hydrogen ions through it, and you could see it rotate like a clock, so they could see. I mean, this is a molecule that's so small. This was quite a ways ago. So cool molecule that rotates like that. And what happens with this ATP synthase means it makes ATP from ADP, right? ADP has 2 phosphates. ATP, or triphosphate, has 3. So it puts a phosphate onto ADP to recharge it. And that happens in this molecule, ATP synthase. As hydrogen ions go through it, it turns and it puts a phosphate on ADP, and it recharges it. Yeah. So cool. And so, okay, so that's at the end of it. Now, how do the hydrogen ions, why the hell are they going through the ATP synthase? Why are they going through it? Because you set up a diffusion gradient, because you use the energy from those electron transporters, the NADH and FADH. You use that like plugging in electricity into the wall. And the electricity travels down and always loses energy as it travels, but it goes through all these proteins studded in that membrane. And each point it loses energy, but it uses it to pump hydrogens, pump hydrogens, pump hydrogens. So you end up pumping all these hydrogens on one side of the membrane. You're concentrating them. And remember, if you're concentrating, like you're going against diffusion, right? It takes energy, because diffusion is high to low concentration. Here we're going from low to high. So energy from that, and the energy comes from burning the sugar and getting the NADH and FADH. And here you use it to pump hydrogens. And so it's like a water tower where the city pumps the water up into that tower, and then it has that energy that can be used. So you're pumping those hydrogen ions on one side, and then they want to get through the membrane, but they can't. They can only get through that ATP synthase channel. And when they go through, just like a current running through your toaster, it can cause work. And it's going to cause ADP to be charged to ATP. All right, how you guys doing? I'm going to run down. And for some of you, I know this chemistry, I may lose some of you somewhere in there. You can read the chapter. If you want to slow it down and just think of it that way, you can rewatch this. Or feel free to give me an email or something like that. I can go through it individually with you too. All right, just beautiful. I mean, this is electron transport chain. Oh, and so what you see is that you're pumping hydrogen, pumping hydrogen, pumping hydrogen. And so there's lots of them out here. And it wants to get from high to low concentration. Well, it can't get through that membrane. It can get through here though. This big, looks like a hot air balloon upside down. And that's gonna turn and it's gonna make ATP. Remember what's called? ATP synthase. It's synthesizing ATP. And what powers it are protons going through the middle of it, hydrogen ions. And it's gonna cause it to turn, it's gonna charge up your ADP. Beautiful. Oh, interestingly, yeah, talk about cyanide. If, how do I say this? Well, if let's say you're not getting along with a spouse or a significant other, and all of a sudden, your coffee tastes like almonds, maybe your cottage cheese, a little almondy. You should be careful. I watch a lot of crime shows too, but cyanide is deadly. It's a deadly poison, and it has an almond taste to it. And why is it deadly? Well, what it does is it goes in and it just screws up one particular molecule, the cytochrome C, cytochrome oxidase, and it screws it up. Damn it, you need that, because if that doesn't work, things stop. And so your cells can't make oxygen. Damn it, they can't make ATP, enough of it, right? Because everything is backed up. So we're talking a small amount of cyanide. If it diffuses in your body, gets to your mitochondria, does that. And don't fear eating almonds. The almonds you get at the store have been the outer part has been taken off, and there's no danger with almonds. But if you were to get raw ones, it'd be a problem. And I hope you guys get this: is that without oxygen, it's the last step. It's like the ground. If you guys are into electricity, at the end, you need that to cause the electrons to flow. And if you don't have oxygen, the flow stops way up here, way upstream, because nothing's getting through. And as soon as you plug it in again, the flow can continue. So when oxygen stops, let's say you're doing a sprint, you can't breathe enough, get enough oxygen, then you're just burning sugars up here with glycolysis. All right. Well, we don't subsist on glucose, right? Yeah, we eat very little glucose, but your starches, your bagels, you convert those starch into glucose when you absorb them. And also fats and proteins, they enter this at different levels. A lot of it is acetyl-CoA. But realize, we talk about so simplistically as glucose is the beginning, but you can have other molecules that enter at different levels and they still work, they go. An interesting thing, I want you guys to think about this. Why do cancer cells favor glycolysis as opposed to using oxygen for a full-born aerobic respiration? Well, the deal is this: cancer cells grow quickly, tumors, and they often don't have enough blood vessels. And so by necessity, as the tumor grows, there's not much blood supply in the middle, and those cells would die if they couldn't do glycolysis. Yeah, and so a lot of cancers, they have molecules, angiogenesis, like make capillaries so they can grow and get their oxygen and nutrients. So you'll see a lot of cancer cells just because they're successful, it's natural selection, all this, they overexpress glycolic enzymes. And so you can find them with PET scans, things like that. Yeah, you can see where are the glycolysis enzymes really being expressed, and they'll light up in the body where they are, and that gives you a hint that it's probably a tumor given off that. All right, when we get too much carbs, you know what happens, right? You have almost unlimited ability to store fat. You can turn any carbs into, into fat. And then the carbs that we eat at a meal get stored as glycogen in between meals. Talk about carb loading. You've got glycogen in your muscle cells, right? That's where you store energy. It's a whole bunch of glucose chained together. That's where we can store some energy. Fat is long-term storage and is denser. Yeah. And then, of course, your liver can do all kinds of things. You can take carbs and you can make a lot of the amino acids, not all of them, by just putting different molecules together. Yeah, but basically, sugars come in. Remember, catabolism is breaking it down for energy. Or when your input is higher than your output, you can store it. Glycogen is temporary. Fat can be more long-term. All right. Wonderful. I front-loaded this lecture a little bit because, yeah, because I'm gonna finish talking about a little bit about a DNA. So we're gonna shift gears a little bit. So you can pause it. Feel free to go get a snack whenever you'd like. And then I'm gonna talk a little bit about DNA. I'm not really gonna wait for it. All right. So DNA, of course, beautiful. We're going to talk about the rest of this chapter is looking at how you turn the genes in your DNA into proteins. And of course, you know how valuable DNA is to today to criminal justice. And who's your daddy? You know, paternity, things like that. So you have, and then of course, this is looking at people before DNA, not that long ago. So many people were wrongly convicted. Because DNA doesn't lie. I mean, it can be planted, things like that. But if your DNA is unique to you, and an identical twin, but even then, not identical. So, and I want you to know when we like compare DNA, we don't sequence 3.5 billion letters. We just get like a dozen or so of these variable areas. So you don't have to sequence it all. You can sequence it all, but there's no need. We know that there are certain areas that vary quite a bit, and nobody of the billions of people on Earth are going to have the exact differences there. It can all be figured out statistically. So DNA contains all the instructions to make all the proteins. And you've got like over 20,000 genes. It can make over 200,000 different proteins because some genes can be tweaked or takes parts of it to make that many products. And the proteins are the skin, keratin, your hemoglobin, insulin, growth factor, antibodies, I'm thinking about things like that, sure. And this DNA is copied faithfully in every one of your cells that goes through mitosis. And of course, your sperm and eggs, you actually divvy out half of it, right? And the sperm, half of the egg. All right, and of course, Watson and Crick, if you read, I forgot which one it was. One of them's kind of an ass. Not kind of, it is. He's sexually harassed a lab mate and believed in eugenics. He wrote things like rich people should be having more babies to improve, that's eugenics is, and Nazis are into that too, the DNA. So again, people like this, and they kind of didn't give credit to Rosalind Franklin, who a woman in the lab. It's the 1950s, and hopefully we've come a little bit farther now, and then things like sexism like that. But yeah, I digress. But anyway, these two, I mean, it's pretty cool. What they discovered is that just from knowing the proportions of the molecules and looking at a couple lines of evidence, they figured out the double helix of DNA. And indeed, the outer parts of this ladder are uninteresting. It's just sugar, phosphate, sugar, phosphate. But the inside, the nitrogenous bases are the A, T, C's, and G's, and that contains the information. So here's a nucleotide. It's going to be sugar, phosphate, and one of these four bases in DNA. And I could ask you, just remember A with T and C with G. Yep. And then these are covalent bonds on the outside, and these are hydrogen bonds in the middle. Yeah, and it's information. our alphabet has 26 letters, right? Computers use 2 ones and zeros. Here we use 4 to build all of us. So pretty cool. And once we figured out the structure, we kind of figured out how it replicates, because you can just unzip it down the middle and you got these 2 halves. And because there's always a complementary base, like A with T, C with G, you can unzip it in the middle and just stick on the correct letter that goes with the other letter. We call it semi-conservative because when you make it, you conserve half of it and you make half new. Yep. So you can see the red is the original. So one of the strands will be the original and one will be brand new. And I don't have time to go through this. You know, it goes in opposite directions. The DNA is unzipped by this DNA helicase. Helicase, anything ACE is an enzyme. It's going to break it apart. And DNA polymerase is going to polymerize or make the DNA. It's going to put the appropriate letter. So if it's a T, it's going to put an A. If it's a C, it's going to put a G. If it's a G, it's going to put a C, right? And it does it amazing. And it has proofreading, and it makes very few mistakes. So DNA polymerase looks like a hand. It goes down like that. Oh, I remember it was a different college, but there was some controversy over this T-shirt. Was it too risque, you know? But DNA helicase, unzipping genes. Anyway, a little biology humor, lighten things up. All right, so your genes. Your genes are just parts of the DNA, locations. And they can be thousands or hundreds, usually thousands to tens of thousands, even 1 million letters, you know, that can, and they're gonna code for a certain protein. And all life uses the same freaking system. I'm looking at 3 letters, these triplets, and coding for specific amino acids. Yeah. And you may say to yourself, I know I seem different than a fruit fly, but how different am I than the guy next to me, you know? Just 1/10 of 1%. Yeah, you think are we 99% the same? Me and a friend? No, 99.9% identical. So that little percentage, remember it's 3.5 billion letters, so it turns out to be a lot of letters, but that's what differs. And actually us differing from chimpanzees is a small fraction, a tiny bit. And most of that difference is just junk, it doesn't matter. So those small differences make the difference between me having a laser pointer and being less hairy, kind of hairy now, than a chimp. Wow. So our genome is all of our genes, all of our DNA. It's found in the nucleus, right? And then ours are in chromosomes. They're little pieces that are around proteins called histones. We have 23 pairs, 1 from each parent. And then the gene will be the sequence. So it's average 3,000 base pairs, 3,000 letters. But they could be tens, hundreds of thousands. And then we read those genes once we clean them up a little bit, edit them into amino acids that make our proteins. Now in prokaryotes and bacteria, it's a little different. Their DNA is in a circle and they don't have a lot of junk. They're kind of straight to the business. Us, we have lots of areas that are just nonsense. And you may say to yourself, 23 pairs, what does that mean? Is that like a magic number? I can look in the stars and figure it out? No. That's just what happened. You can see chickens have 39, goldfish have 47, fruit flies have 4. I mean, it's how you divvy it up. As long as you know where the recipes are, it doesn't matter how many pieces you have. It's just, it kind of came that way in evolution and we've inherited that. And then, of course, alleles are different versions of the gene. So blue eyes, you might have brown eyes. That's not a single gene, but we have genes for hemoglobin. And we have early on, we made copies of them and we altered them. So we have fetal hemoglobin, adult hemoglobin. We'll get to that too. But alleles are different versions. Yep, and they allow us this beautiful variation. And evolution can work on these alleles. If one is selected in environments, you're gonna have more copies over time, right? So as I said, you can take one cell out of my body and it has all the genes to make a whole new Jeff. All the information is there. Yeah, and then we're talking about amount. Fine, we'll get to the last slide. The amount of DNA. Well, it turns out, especially in us, a lot of our DNA is junk. So you see, how many of those letters, 3.2 billion, code for genes? Well, if you're thinking to yourself, we have like 20,000 genes. Oh, shoot, we're only like 3,000 on average. where's the rest of those letters? What are they doing? Well, 98% of the letters in our DNA appear to do nothing. They are long strings of A's and T's that get copied each time. So it's a waste of time, but we copy them. So why do we have this burden? You can see a bacteria, only 10% is wasted. They use 90% of their letters. Oh, this is a great question, and we could discuss. One thing is it's hard to get rid of DNA. So once you build up this junk, it's hard to get rid of it. So just like as you get older, you keep accumulating stuff each time you move, kind of thing. Yeah. And also, since 98% of our letters do nothing, that means if there was a mutation at any of those ones, it would not cause anything bad. So it's kind of protection. It kind of hides our genes in these small areas where statistically you're unlikely to get them banged up. All right, so we have a lot of junk DNA that apparently does nothing. And I say apparently because I couldn't tell you that in 10 years from now we don't realize that stuff is doing something, but it seems to be long sequences of garbage. All right, well, we're going to get to how genes work, definitely, especially in the next lecture. But just introduce it, and then we have a lecture that realize that your genotype is all your genes. Your phenotype is what you look like. So it depends on how I cut my hair. I can color my hair. I can shave my head, right? My phenotype can look different, but genotype is what's my genes are going to tell me. And we're a product of both, aren't we? Genes and environment. It's amazing how much is genes, though. You get identical twin studies that are raised in different families. So many things we're just guided by our genes. So I swear this lecture about over, but this is, I'm going to start with the next lecture, looking at how this works. And I've told you this before, but your genes are kept in your DNA and your nucleus where they're safe. They're in a vault. And then you're going to open up the DNA and read it. It's called transcribing. Like a transcriptionist in a court says everything that you say. It's going to copy that recipe. And that recipe, we call it messenger RNA, is going to go into the cytoplasm. So it's like the recipe on an index card. And then out there, we translate it into the protein, into the recipe. Yeah, and so the messenger RNA goes through these ribosomes, and then protein comes off the top based on that recipe. Yeah. One little change, you add raisins. I don't know why I want to do that instead of chocolate chips, but little things like if you have a mutation over in your DNA, it's going to show up in your recipe. In many cases, it doesn't make a difference, but sometimes it does, right? All right, finally, yes. So your genes contain all of the recipes in your body to make all your proteins. Yeah, very cool. All right, class, I made it through. Again, longest lecture, so I hope you feel free to pause in there. You don't need my permission, do you? So I'll have one more much shorter lecture to finish up this chapter. I put some sample exam questions up there for you, like 40 questions. You can take that. You won't be ready for it now unless you've finished this last chapter. And I hope you're all well.